Dear colleagues, my name is Professor Natalia Linkova. I am the head of the Laboratory of Molecular Mechanism of Aging of St. Petersburg Institute of Bioregulation and Gerontology. The theme of my lecture is Peptide bioregulators application in cosmetology, clinical and molecular aspects. The reason of skin aging is exposure to ultraviolet radiation, sunlight and stress. So we can see that skin aging correlates with violation of neuroimmunoendocrine function of skin fibroblasts, violation of collagen synthesis, metabolism and divisions of skin fibroblasts, and violation of immune skin cells function. For normalization of all these parameters, we can use peptides. Peptides regulators of neuroimmunoendocrine skin function, peptides regulators of fibroblast skin function, and peptides regulators of immune skin function. Endolutein is polypeptide complex from pineal gland, the regulator of the neuroimmunoendocrine system and geroprotector. It was detected in the polypeptide complex from pineal gland by mass spectrometry and high-performance liquid chromatography. Free amino acids D, free tetra and pentapeptides. The IEDG peptide was identified among tetrapeptides in pineal gland polypeptide complex by the method of selective monitoring of reaction. So, the IEDG peptide is an active component of endolutein. The IEDG peptide regulates circadian rhythms, function of the endocrine and immune systems, has an antioxidant effect, stress protective effect, increases the length of telomere in cells, and increases the life expectancy in experiment. Since the skin is an organ of neuroimmunoendocrine system, the IEDG peptide contributes to the normalization of skin cells function. Sigomir is a polypeptide complex from cartilage and bone tissues, a regulator of connective tissue cells, cartilage and skin fibroblast functions. Sigomir was analyzed at the Institute of Toxicology of MBA of Russia for the content of the IED peptide Cartalax. So, IED peptide Cartalax is an active component of Sigomir. Endoprotective effect of Cartalax efficiency in osteoarthritis, osteoporosis and other diseases are due to the activation of proliferation cell division of cartilage fibroblasts. Fibroblasts have similar properties in all organs and tissues. Cartalax has a stimulating effect on the metabolism of skin fibroblasts. Vladanix is a polypeptide complex from thymus, a regulator on the functions of immune system. Vladanix was analyzed at the Institute of Toxicology of the FMBA of Russia for the constituent of the EDP peptide cristagen. So, EDP peptide cristagen is an active component of Vladanix. The EDP peptide stimulates differentiation and activation of B cells, macrophages, proliferation of immune cells during aging, reduces apoptosis of stem and immune cells during aging. At the systemic level, those effects of EDP peptide are expressed in regulation on immune system functions, antioxidant action and stress protective effect. The skin performs a protective barrier function and contains a large number of immune cells. Vladanix and its constituent, the EDP peptide cristagen, can contribute to the normalization of the function of immune skin during aging. Peptide bioregulators application in cosmetology study design. The study was conducted on the basis of the Anti-Aging Medicine Clinic of Media, St. Petersburg. There were 15 middle-aged and 15 elderly women in each group. To assess the effectiveness of peptide therapy, we use the questionnaire Dermatological Index of Quality of Life. Also, it was used the assessment of the thickness, homogeneity of the dermis and epidermis of the skin by ultrasound scan method. The first group control is a saline solution application by electrophoresis on the skin area once a week during 8 weeks. The second group is application Peptides, Cartalax and Cristagens on the same scheme as in the first group. And the third group is the Peptides, Cartalax and Cristagen application by electrophoresis on the face area once a week during 8 weeks. Women from this group received oral polypeptide complexes Sigumir and Endolutein. So we can see the influence of peptides on the skin condition of different age women according the questionnaire. The maximum effectiveness was observed in middle-aged women in both investigated groups. So this is the influence of peptides on the thickness heterogeneity of the dermis in a middle-aged woman. We can see that before treatment, derma was thick and epidermis also was thick, and after the treatment, in the third group, when we use application of electrophoresis with peptides and peroral peptides, Sigumir and Endolutein, we can do that the level of derma and epidermis increase. So we can see that application of electrophoresis with Cartalax and Cristagen in combination with Endolutein and Sigumir in capsules contributed to a 20% increase the thickness of dermis in middle-aged women. Also, we can see that application of short peptides and polypeptides complex contribute to an increase on 15% the thickness of epidermis in middle-aged women. The same experiment we do on the skin of elderly women. Before treatment, we can see thick dermal layer and thick epidermis layer, and after we use short peptides and polypeptide complexes, we can see the increase of derma and epidermis parameters. Single application electrophoresis with Cartalax and Cristagen, and its use in combination with Endolutein and Sigumir in capsules increase on 19 and 38% the thickness of dermis in elderly women. Application of electrophoresis with short peptides and polypeptides complexes in capsules increase on 15% the thickness of epidermis in elderly women. On this slide, we can do the molecular experiments on the cell culture from 35 years old woman. And here we can see the immunofluorescent confocal microscopy of skin fibroblast. Investigated skin fibroblast cultures was divided into 6 groups. The first group is control intact cells. The third and the second group is control peptide GGG and experimental peptides IDG. And 4, 5 and 6 groups is fibroblast under the action of on ultraviolet radiation, the model of skin aging. We can see that IDG peptide activates SOD1 gene expression in the model of skin fibroblast accelerated aging. Superoxide dismutase 1 or SOD1 is an antioxidant enzyme that protects the intercellular space from superoxide anions by catalyzing their conversion into molecular oxygen and hydroxygen peroxide. SOD1 gene expression decrease in human skin fibroblasts during aging, and IDG peptide increase SOD1 gene expression by 3.5 times. Also, the IDG peptide activates catalase gene expression in the model of skin fibroblast aging. Catalase is an enzyme of the oxidoreductase class, which is part of the antioxidant system of the cells. It performs the function of antiperoxide protection. This enzyme catalyzes the decomposition of hydrogen peroxide formed during biological oxidation into water and molecular oxygen. Also, IDG peptide activates NQO1 gene expression in the model of skin fibroblast aging. And we can see that IDG peptides activate the expression of this gene also without ultraviolet irradiation. NQO1 is quinone oxidoreductase enzyme is localized in the cytosol inside of the cells, catalyzes the 2-electron reduction of quinone compounds and prevents the formation of semiquinone free radicals and active oxygen molecules, protecting cell from oxidative stress. On this slide, we can see the results how Cartalax and IDG peptide regulate protein synthesis in skin fibroblasts during aging in culture. So IDG peptide have geroprotective and antioxidant effect. The increase of sirtuin-1 and 6 protein. All the IDG peptides stimulate collagen-1 type synthesis. IDG peptide have anti-apoptotic effect. It decreases the expression of P53, P16 and caspase-3. All these peptides have proliferative activity and increase the synthesis of Ki67 and CD90H and C proteins. Also, these peptides can regulate extracellular matrix remodeling by regulate of MMP9 expression. On this slide, we can see the control group. Green fluorescence is collagen synthesis in skin fibroblast. After we added the IND3 peptide, we can see that the synthesis of collagen-1 increased, so we can see that the level of green fluorescence was high. On this slide, we can see the expression of nuclear protein Ki67. This is the pink fluorescence in nuclear cells in control. And after we added the IDG tetrapeptide, we can see that the number of Ki67 positive cells increased. So the pink fluorescence was more intensive. This is the scheme of peptide regulation of skin function. The IDG peptide, an active component of endoluten, the IED peptide Cartalax, an active component of Sigumir, and EDP peptide Cristagen, an active component of Ladonix, can interact with DNA. So we can see that this peptide regulates gene expression and protein synthesis, regulation in skin fibroblast. So it is the normalization of the thickness of dermis and epidermis in skin during aging. We can do the conclusion. Polypeptide complexes of cartilage, sigumir, pineal gland, endoluten, thymus, and IDG peptide Cartalax, EDP peptide Cristagen increase the thickness of the epidermis and dermis in middle-aged and elderly women, according to the questionnaire and evaluation of the thickness of the epidermis and dermis by ultrasound diagnosis. This helps to slow down the aging of skin cells and improve its appearance. Cartalax and IDG peptide interact with promoters of gene-according proteins of functional activity of skin fibroblast. This contributes to the normalization of skin fibroblast function with their natural and accelerated aging. The obtained data indicates the effectiveness of peptide bioregulators Sigumir, endoluten, Cristagen, and Cartalax to slow down age-related skin change. Those peptide bioregulators can be used to improve the quality of the life and satisfaction with their appearance in middle-aged and elderly women. Thank you for your attention.